design to mechanistic understanding, and computational tools such as molecular docking and virtual screening, quantitative structural activity relationship, popularly called it as QSAR modeling, and molecular dynamic simulations enable rapid identification and optimization of potential anti-cancer agents with high precision and reduced cost. These methods facilitate the prediction of drug target interactions, binding affinities, and pharmacokinetic profiles, providing valuable insights into molecular mechanisms and resistance pathways. By combining bioinformatics, cheminformatics, and AI-driven modeling, in silico approaches accelerate the transition from hit identification to lead optimization, thereby enhancing the efficiency and success rate of the anti-cancer drug discovery. On behalf of VV Institute of Pharmaceutical Sciences, Gudlavalledu and APTI Central Bangalore, I extend our sincere gratitude to Dr. Y. Prasananjan garu. Prasananjan garu is having a very good, vast research experience, okay, in this particular in silico molecular modeling. I hope that today's deliberations are very much useful for the academic benefit of the faculty members. Thank you, sir, for graciously sharing his valuable time and expertise for the academic enrichment of our faculty members. Thank you all for your enthusiastic participation. On behalf of the institution, I thank you all for the active participation. Now I invite Ms. M. Harika Garu, convener of this FTP, to proceed over the program. Over to Harika Garu.
Thank you, sir. Now I invite K. Shanti Priya, Assistant Professor in BBIPS, to formally introduce today's distinguished speaker, Dr. Y. Prasanna Anjaneyulu, sir.
Good morning to Good morning to the respected principal sir and our speaker, Dr. Prasanna sir. Dr. Prasanna sir is a
Yeah,
medicinal
yes, good morning.
chemist specializing in structure and ligand-based drug
Hello.
design.
Hello.
Hello.
Madam, you continue.
Dr. Prasanna sir is a medicinal chemist specializing in structure and ligand-based drug design, cancer drug discovery, and plant metabolics. He completed his B Pharmacy in CHIPS College, Gunturu in the year 2017, securing top ranks in the graduate pharmacy aptitude test with 777 air and NIPER with 297 air. And subsequently, he earned his MS in medicinal chemistry from the National Institute of Pharmaceutical Education and Research, Ahmedabad, in the year 2019. He joined Jamia Ahmad, New Delhi, as a junior research fellow under the DST BRICS collaborative project, later continuing as a senior research fellow while pursuing his PhD in pharmaceutical chemistry from 2020 to 2023. His doctoral research focused on designing and synthesizing novel N-heterocyclic conjugates targeting cancer-related pathways, including P13K trankinase and VNT signaling, integrating molecular modeling, SAR analysis, and preclinical in vitro and in vivo studies. Dr. Prasanna sir has authorized over 18 publications in high-impact journals, including ACS Medicinal Chemistry Letters, Organic Letters, RSC Medicinal Chemistry, and Expert Opinion on Drug Discovery, and has presented his work at national and international conferences. He received the DST BRICS Travel Grant for collaborative research at the University of the Witwatersrand and the University of Johannesburg, South Africa. In 2023, he gained industrial experience as senior research scientist at PI Industries Limited India, contributing to preclinical small molecule discovery. Currently, at the Ohio State University, he is engaged in the design and synthesis of iron chlorophyll derivatives for transitional studies supported by the Bill and Melinda Gates Foundation and Abbott Pharmaceuticals. Beyond research, he serves as Vice Chair of the APS FarmSci 360 track, San Antonio, and as peer reviewer for reputed scientific journals. Thank you, sir.
Yeah, thank you very much. So for letting me know by bio and everything. Yeah, shall
Yes,
we
sir.
start the
Now I request Dr. Prasanna Anjaneyulu sir for your engaging and highly insightful lecture. On behalf of VV Institute of Pharmaceutical Sciences, we express our deep gratitude and uh, and continue this section, sir. Thank you, sir.
okay, ma'am. Thanks for the introduction and everything, and thank you each and everyone, principal sir and Harga ma'am, everyone. So thanks for introduction. So now I'm starting my lecture.
Yes, sir.
Is it visible, ma'am?
Yes, sir. It is visible, sir. Please put
Okay.
it in a slideshow mode, sir.
Yeah,
On
yeah,
full screen, you
yes,
will
sir.
get it.
Yeah. Okay, so all we know, this is the Prasanna Anjaneyulu Akala. Yes, I am working as a postdoc in the Ohio State University. So here I am delivering the integrating in silico approaches in anti-cancer drug discovery from molecular design to mechanical understanding. I means I will I will let you know from where is the scratch and where is the ending of the this designing of molecules and everything. So here I am giving the overview. How do we design the molecules, like the overview of the work. So this is the basically I am combining the two works, and one is sponsored by the Bilgers, and another one is sponsored by like one of the past works sponsored by the DST BRICS project. DST BRICS is a 5 countries collaborative project like BRICS, Brazil, and this thing. Okay, here uh, we design some molecules based on the structural design, structural based drug design, and we choose some proteins and we go for and dock the molecules with the protein, and after the docking and everything, so what are the molecules have the good docking score, free energies, and so we screen optimize those molecules and we test the molecules. We call have molecular hits, and after that it is not possible to go with each and every molecule. Some of the molecules are discarded based on the docking scores or bad poses on bad interactions, and they are not fitting in the binding pockets. So based on the criteria, we discard some of the molecules, and after that, we passes through the pain filters. So pain filters is one of the criteria. The molecule should have the like 5 criteria: Lipinski rule of 5, and the molecular weight less than 5, and the lipophilicity, and hydrogen bond acceptor donor. So based on these things, we should filter some of the molecules, and after that, although we filter some of the molecules is very hard to synthesize the synthetic feasibility. So based on the criteria also we choose some of the molecules, and after that we go for the in silico ADME, like the pharmacokinetic and pharmacodynamic properties of the molecules, and after that we select the library of the compound. So after that we synthesize the compound. Let me have the pointer. So after that, we synthesize the molecule. After the synthesizing molecule, we go for the MTT assay. MTT assay is nothing but cytotoxicity assay. So we have the different cell lines. Like for the right now, I'm focusing on the colorectal cancer. So we have different cell lines like CACO2, HT116, and BLCL2. So a lot of cell lines are there. We try all the cell lines, and after the trying cell lines, the molecule which showing the potent to have activity will go for the in silico in vitro target studies, like the Western blot and the studies, the gene expression. And after that, the molecules which are more potent and active, we go for the mice model, and the ultimate final molecule will go to the clinical trials. This is the flow of how the molecules will work. So now we go for the step by step, and like the in silico designing is like a huge topic. So now I'm like it is not and now I'm taking the only one example and explaining how we targeted the all the approaches. So here in silico design and validation of the paired in based to 1 to 4 triazoles that are Hindole conjugates for the TNKase means tankerases, and PI3K means phosphatidylate 3 kinase inhibitors. So here we are targeting the both TNKS and PI3Ks. It is the interest from the DST Bricks project. So we got the funding from there. That's why I worked on this thing. So this is majorly seen in the colorectal cancer. Here already I told you. So here tankerases and the PI3K are the target proteins. So I choose this one. And our target molecules is pyridine based 1 to 4 trioles. You people can ask, there are a lot of molecules can act as a PI3K and tankerase. How can you know these pyridine 1 to 4 trioles as the indoles as the only lead molecules? I'll explain that thing also. Like, why do I choose this thing? Although we have the indoles, trioles, naphthoquinones, tetrazoles, naphthenols, benzodiazole, lot of n number of molecules are there. Why I choose only these molecules apart from the all the molecules? Then earlier I told you we will go for the molecular docking and we go for the molecular screening, and these are the different various parameters by optimizing the lead identification. So before going to the each and everything, before going to the cancer, like I know what is my target. I mentioned that's a PA3K target. So first of all, I need to see the what are the different different enzymes. Different enzymes are pathways presenting in the PA3K pathway. So among them, it is like a structure-based drug design. I told you. So here. So based on the PA3K pathway, what like every protein has the internal ligand. So ligand is nothing but the like chemical structure. So in the ligand, I can see the if ligand based degradation means we see the ligand, and based on the ligand, we can go for the similar type of the designing of the molecule. So what I did, I choose the PA3K pathway. If you take the PA3K pathway, you get the n number of proteins. There is one data bank that is called protein data bank. If you open the protein data bank, you can see all the PDBs present in the protein data bank, and we have to select the protein which has the resolution and the free R square value energy and R factor and the hydrophilicity. So a lot of criteria and parameters are there. Based on the parameters, we choose some of the proteins from the library from the protein data bank, and after the picking the protein, we visualize the protein by using the Schrodinger software. So I use the Schrodinger software. Apart from the Schrodinger software, you also have that Autodeck and Autodeck Vena and lot of softwares are there. So I use the Schrodinger software, and where I able to see the designing of able to see the molecule present, ligand present in the protein. So based on the ligand, we take the structures. So the structures more or less type of like combination of the naphthquinone and pyridine and type of indole and a few type of other heteromolecules. It's like a huge complex molecules. So it is not possible to binding all like combining all the structures. That's why we go for the each and every fragment on such a way. So here this is the pathway, like how the PI3K pathway are there, and based on the pathway, just I go for the PI3K protein and I choose from the this is the protein. So as per the literature, like before going also protein, I would like to cross check with the literature. What are the molecules are represent for the PA3K inhibition activity? So these are the various molecules with having the IC50 value like 1.3 micromolar. IC50 value is nothing but the activity, how much potent of the molecule based on the target. So this contains like a naphtha type of molecules are there, and indo type of molecules are there, and normal naphthenol type of molecules are there. So different type of molecules are there. So based on the molecules, we have the IC50 values. So I'm confirming that what I am targeting is really matching with the existing literature or not. So similar way, I also take the tankerase pathway and I understood what are the different proteins in the same way to how the applicable for the PA3K inhibitors. So here also I did the same thing. I did a survey and I found some of the molecule. This type of molecules are what we call as in clinical trials. Some are has a great in vivo and in vitro potential activity. and few are the very good for the like these biological properties and the ADME properties and everything. So now I felt okay, I'm in the right tag. So exist literature also have the this type of molecules, and I am also doing the same type of molecules, and I am now I can good to go for the thing. So finally, I go for the dual inhibitors. Like I am combining the both the pathways, how the PA3K is coming and how the tanker is coming, and how they are junctioning on the protein exactly. Because now here I am going for the two targets. One is PA3K, one is tanker S. Is like one left hand is for the PA3K and the right hand is for the tanker S. My molecules, one part will go and bind the PA3K, one part will go and bind the tanker S. So this is our ultimate target. And I also got the few literature. These type of molecules are well known for the both PA3K and tanker areas. So now based on the molecules and based on my interest, I choose few molecules. Among the molecules, I rationally design the somewhat. I rationally design the this type of the scaffold, not the exact molecule. So based on this type of scaffold, here instead of the indoors, we also tried with the naphthoquinones and benzothiones and this thing, benzothiones and like naphthenols and anthracene. Lot of molecules over here as per literature. Here also we did a lot of modification, and all the modification we have around like 250 or 300 molecules. I will show the all the like it is not possible to show over here. I have the backup one. I will show that later. So all the among the 150 or sorry 250 or 300 molecules, I need to like it is not possible to go for the each and every molecules and go and test in the lab. It is very hard to me. That's why we choose the molecular docking and the molecular docking are in silico varieties to optimize few of the molecules. So as I explained you, like if you see over here, this is the molecule of my target. Here, if you see the N substituent over here, it is the like oxyazole, and same way here I go increase a naphthoquinone, and here is a triazole, and here is like internal derivatives, and here is like different carbon derivative. So and naphthalene derivatives. So we go different type of these things. Just like mentioned over here, functional groups. Apart from the functional group, I used to keep the R. So actually there is some database in the websites like PubMed or like Zinc database. So if you see if you the scaffold what you are designing, go and keep the scaffold over there. Once you keep your scaffold, you'll pop up the lot of n number of molecules which is like which is some are reported and some are non-reported. So based on those molecules, you have to like focus which type of molecule are there, and second thing, are there anything work are done on these molecules or not, and the third thing is it is done what is the those values, and one more thing, if it is a new or novel, then we go for again we go for the SciFinder and we check the molecules whether the molecules are the like properly novel or not. So based on the thing, we go for the next studies. So here I prepared all the 250 molecules, and I mentioned you, I go for the molecular docking. So here molecular docking, it is like just here I only mentioned the 3D in ribbon shape. We have the binding pocket, and like if you go for the binding pocket or socket type, so it is very hard to visual. That's why I go ahead only for the ribbon type of the molecules. So once the molecule in, I have to check how the interactions are going on, like is it hydrophobic interaction, hydrophilic interaction, ionic bond interaction, or van der Waals forces, or like salt bridges, or like all the different type of interactions, how it is showing, which type of amino acid is showing. Is it like how the previous molecules are shown the same way, or am I seeing something is new? So because the protein is the huge structure, if you open the protein, you get the n number of sites and n number of long chains. So it is very hard to find the exact set. It is like a, how can I say, like, yeah, searching needle in a hashtag, like a lot of molecules, and we were searching it like a small binding pocket with a small molecules. So based on this thing, we have to like we have, that's why we keep some parameters. So like the checkout parameters, yeah, my molecules is fitting this thing, so I can go past the next level. It is not fitting, I will stop the next level. So based on thing, I used to get around like 60 or 70 hits or something other molecules, because some of don't have the interaction this thing. So after that, so here I also check the docking score and the like the bad poses. Bad poses means the molecule is not going and fitting to the binding pocket. Only some words of the molecule exposing to the protein, and some are getting the outside of the protein. So that is called bad poses. That's why I'm not interested in those poses. And the like the molecule is then the docking score is like how much efficiently the molecule will go and dock or bind means docking score. If you go and efficiently bind with the protein with the like high, then its docking score will be the high. Like that will be representing the minus like minus 10 kilojoules and whatever that this thing. So based on the docking score, we optimize the things. And the pain filters. So in the web in the Google, just you type there like pain filters, so you can get the pop up the website. And after the synthesis molecule, just you upload over there and you click on the pass. So after that, the really the pain filter is like the molecules are like druggable or the some I told you earlier a few criteria are there. If each and every criteria will passes, then the molecule will pass from the pain filters. So the molecules which are passes from the pain filters are only the molecules acceptable for the clinical studies are in vitro or in vivo. If it is not, so it is not acceptable. So that's why we pain filters should be necessary. Then after that, a synthetic feasibility. So the molecules which is feasible in the lab, I told you that I use the same thing. And in silico ADME. ADME means like we have the absorption, distribution, and elimination and whatever. These are the one more few more criteria we should choose to the molecules, which includes like Lipin's rule of five and hydromorphs acceptor, donors, and molecular weight and permeability and everything. So based on the thing, we choose select the molecules. So here, although I mentioned I worked on like 250 molecules or something, so we able to see only the like 3 or 4 molecules as a lead molecules. So although I'm not sure, it may be lead, may not be lead. Until and unless it was done, then only I confirm that these are the leads, or else these are not the leads. So apart from also, we also did the some QSR parameters, like QSR studies and a lot of studies to cross check all these parameters. What I am designing molecules, whether is really the thing what I'm expecting or not. But it will be getting the huge data. That's why I'm like concising only few things only. Just I'm explaining the outline what's going on here. So these are the different like. Although I have the 250 molecules, I am only representing the 17 molecules only. So in the 17 molecules, I showed you the 3 lead molecules. Like why should I choose the 3 lead molecules? Because of the like the glide score or glide energy or GIPSA means like the free binding energy, how the molecules are freely interacting and everything, and how the molecules are interacting with the Tankerages and how the molecules are interacting with the PI3K. So these are the type of the hydrogen bonding which I able to see, and I also cross checked with the literature how the molecules are binding to the bonding to the interaction. So we get the similar type of interactions. And like I told, the docking score is around like for the Tankaris is minus 9.7. If you have the greater like lower values like minus 9, minus 10, minus 12, minus 13 means it has high efficiency for the binding. If it has like 5 or like 4, 6, it means it has the low efficiency for the binding. For it's both PA3K and Tankerages, it seems like Tankerages are the more efficiently binding to the pocket when compared to PA3K, because in PA3K we have the very like low binding score like 6 and something, and although we have the binding score of 4 also 3, so when compared to these guys, so the 14 is acting as like better from the rest of the molecules. So here one of the criteria is I completed, and after that I also don't know or really those molecules are based on the docking score. I can conclude that although I did the QSA parameters and I also did the docking studies, and based on the both parameters I able to screen only those 3 molecules, but it is just not a like validating of the molecules. That's why I go one more step. It's called molecular dynamic stimulations. So in the molecular dynamic stimulations, we go for the different time intervals of the molecules, like what it will do exactly. So the molecule, it will go in the binding pocket and fit in the binding pocket, and we give the 10 nanoseconds of like simulation or 100 or 50 or like different type of stimulations. So it will give n number of the uh, confirmations, how the molecule will go and show the water molecule the integrity and the binding and the like RMSD value, and put on like interaction, and how frequent they're contacting, is the bonding or the really hydrogenic bonding or like van der Waals bonding or water bridge or whatever, the hydrophobic, hydrophilic. Earlier in the docking also, like in the docking, if you will go and fit inside the molecule, and it may be true, may not be true, because there is only one confirmation, is like the how can we just we like we keeping the key in a lock. Yes, you go and it's done. We don't know whether it's working or not. But in the MD simulation, it's like, yeah, we are rotating the key inside the lock. It means we are checking it whether it's really fitting inside the lock and is opening the key or not. Some of the keys like have the similar properties, will go and fit into it, but it can't open the keys or open the lock. So same way, the like the molecular docking is like just inserting the key, and dynamic stimulation is like opening of the key. So we did all the parameters, and yes, yeah, fortunately we got the very good results of the protein like RMSD, RMSF and RMSD, and protein like interaction. So what I'm expecting is I'm getting the same thing over here. Then I concluded, okay, yeah, I am good to go for these molecules. Similar way, as I mentioned you, like the molecular weight should be go less than 500, and the log pre property will be should go for the like higher one, and hydrogen bonus should be less than 5, and acceptor should be less than 10, and the SAS is the like the one type of the energy, so it should be the higher is good for the molecules. And the human absorption, like the absorption should be the 100%, then only it will go for the bioavailability and bioaccessibility of the human bodies. And we are the final lipin rule of 5. If it is the 5 0, then it is a good. If you violating the one of the rule, it means the molecule is not acceptable for the next work and everything. So these are the molecules like I did for the all the like these molecules called properties. So we did the like in silico like MDE property something. So we insert our molecules, and the based on the molecules, it will give pop up the how the molecule like behavior of the molecules and everything. So based on the Schrödinger, we optimize all the parameters. Now I come to know these are the lead molecules. So, but again, till now we have only the theoretical work. Oh, because based on the software, it explained. Maybe the software right or wrong, we don't know. Now we go for the next step, that is called practical. So now, based on the molecule, the lead molecule, I optimize the synthetic cream. So where I do the start from the scratch level, and how do I do the like I go for the like uh, hydroxide and treat with the isothiocyanate and with the ethanol and respect to thiocyanate carbonate. And after that, I go for cyclization and got the triazose. And after triazose, I treated with the 3-bromo indolent, and finally I got my final lead molecule. This is the scheme I proposed at the time, and finally it's worked fortunately. So based on the this library, we derivatize the different type of derivatives what I showing from the top mode onwards. And these are the 3 leads what I found in the based on the docking or everything. So on these studies, then I am going for the confirmation, because once it's done, how can I prove that is really good or not, like what I'm the thinking is done or not. So that's why we go for the NMR. In the NMR, the molecule should be the pure. If it is not pure, it means some of the like the impurities present in the molecule that also maybe causes the activity. We don't know until and unless. So that's why the NMR like the molecule should be the pure. If you see these peaks, these are the peaks. If you see this small gas over here, it is like the impurities. So sometimes what happen, these impurities called the activity, and the major lead molecules can cause the like inactive. It was happening with one of my molecules. The molecule is showing activity, but after that is not showing the activity. Then after that, we conclude that there is a mixture of the molecule present, that is called 95 to 5% of ratio. Those 5% of ratio is getting the activity. The 95% is not getting the activity. So it's happening with my case also. That's why we have to cross check the everything. And after confirmation, just we go for the carbon number to cross check. The proton is the only give the peaks, and carbon give also give the like what are the carbons and chemical shifts and everything. We confirm the carbon number, and we also confirm from the HRMS like high resolution mass spectroscopy and the AR. Here also in every aspect, it confirming yeah this is the molecule what I am looking is the same. So now we are going for the biological properties. So in the biological properties, we take the 96 well plates. So like 96 well plate means we have the square type of plate. We have the 96 wells inside the plate. So we seeded means loading. We loaded the all these cancer cell lines. The CACOT is the colon cancer line, DLD is the colon cancer cell line, A170 is the lung cancer cell line, A154 is the lung cancer cell line, and Tera is the colorectal cancer cell line, ST is the colon cancer cell line. So we load the all the cells like for the each well we load 10,000 cells and we incubate those cells to give the dominant or potential or something. So once it's done, we take our drug molecules like from the 14A to Q and we take the different concentration because we don't know what concentration the molecule will show the activity. It may be the lower, it may be the higher. It will be the sometimes what happen, the cells will die because of the toxicity, not for the activity. So anything will happen. So we don't know. That's why we go for the different concentrations from the lower to higher, like 0.5 microliter to 5 microliter. So we go for the different concentration and we introduce all the molecules into the 96 well plates, and after that we go for the graphite prism and to calculate the IC50 concentration. Like 50 IC50 means like 50% of the cell should be die and 50% should be the add to alive or something. And based on thing, if cells is all are dying, it means your drug is potent. I told you one more thing. It may be the toxicity also, it may be the potent also. That's why we do some control experiments and everything to confirm that these are the like potent molecules. And here also I got these molecules are the very potent and I used to get the like 1 micromolar over here and when H20 cell length and remaining I used to get the like a little bit high, but 1 micromolar is like the one of the type of like a promising molecules. And here I used to get 3.6 2.4. It means like in the H29 the molecule is like almost very potent when compared to the other cell lines. So we take these cell lines and we proceed with the rest of studies. So here it is called cell cycle analysis. My molecule is going and killing the cancer. How is happening? What is the mechanistic pathway is working? So this is called all we know. We have the different phases in the cell, like G phase, S phase, M phase, and G1 phase. So it will go and which way is it is exactly going acting. If you see the color codes over here, these are the different color codes like G1 is a pink and G0 or G1 is the orange and S is the like somewhat like green, like bluish green or whatever, and G2 is the like green. So if you see the intensity of the cell population, so most of the like G1 phase is going and blocking the all the like the cells. So now I conclude that okay, my molecule is going and blocking the only G0 phase. Okay, I conclude the cell cycle analysis. Next, how the mitochondria ROS, like ROS is like a free radical something. So how the mechanical relative is going and blocking the thing. So if you take the ROS of the pathway and it will give you the brief how it will go and attacking and binding the pathway and how we call as with ROS something, but I like apoptosis, like programmed cell death. How it happening? Is that like a priest earlier it was happening or lately it was happening? So based on the conclude that was happening in the late stage early stage apoptosis fifth of my molecules. So if I see over here, the orange gives the late stage and pink gives the early stage. So these are the dye to identify the aggregation, like monomer, how like the dye will go and monomer aggregated are multimer aggregated to kill the cells and everything. So this is also like a similar type of ROAS and which give the or like a population, like how the population seems like after the treating of the drug and cell lines. So it will explain the like briefly how the molecular activity and immunofluorescence assay is like a 3D fluorescence assay. Once I treated with my cells, so after that I really don't know like cancer is like a big lump. So after treating with that dye, the big lump will go into the small lumps. It will divide it, and the lumps will be going to gradually decreases the size of the lump. So this is the happening. So in the immunofluorescence assay, we go for the hello. Is it okay?
Sir, okay, sir.
Yeah. So okay, in immunofluorescence assay, we go for the like the
वही तो वो कह रहे थे, हाँ
I
इवेंट
request
हुआ ही नहीं
every
इतने सालों
participants
से।
to unmute you, mute your speakers.
तो भी कहते मैम कम से कम गवर्नमेंट वाले मजे तो लें।
Okay, sir,
okay.
carry on. Yes.
Yeah. So based on the like we have the control cells over here. After control cells, we used to go for the different molecules that dying. If you see the control is the more intense, means is the like the cancer and the like the cells and everything seems like how the shapes and everything rigidity. And after that treating with thing, how the cells degradation is take place and the size of the tumors and everything. So like based on that, this experiment will be done for the repetitive time intervals like the 8 hours, 16 hours, 24 hours, and based on the time intervals, we have to identify the immunofluorescence assay, how the how is really affecting the pathway and everything. And after that, finally, we go for the Western blot analysis. So in the Western blot analysis, we load the whatever the desire. Like I told you, I block the PI3K and tankerase. Really, yeah, am I block the PI3K tankerase? I don't know. So what I do over here, I now if you go if you remember the plus side, I will mention the all the genes present in the pathway, the APC, auxin, and PAKT, and tankerase, and PI3K, the beta cat, the PI3K is this gene, and TBK is the tankerase gene, and beta Wnt pathway is the like beta catenin gene. So these are the remaining genes which supporting to the things. So some should be the directly proportional, some should be the indirectly proportional. Like directly proportional means if one of the gene is knockout, then the rest of the gene will be knockout. Like if you see the PAKT and PA3K, if you're blocking the gene present in the PA3K, it should be knockout the same way in the AKT because they are directly proportional. If you similar way, if the beta canon also directly proportional. If we're knockouting over here, the expression will be the high over here, such a way. So we load the all the genes and we run the blot and we check the what are the expression of the genes. So here my molecule are like if you see the all the control, the intensity of the blot will be the little bit intense. When we go for the my lead molecules, the intensity should be drastically decreased, like not drastically, like most of the very negligible. So here also for the pan-theatric also for the control, the like the control the gene is high when compared to the molecules. So such a way we identify the pathway. Okay, it is blocking AKT, it is blocking PA3K, it is blocking the beta catenin. The tanker is also blocking little bit, and the GSK also little bit like so. Based on the thing, we can conclude how it varying, how it doing, and how it's going on. So next we go for the SAR. Although I mentioned that QSAR are the like different of molecules, I already go before to the screening and everything, like how the molecules are going and the atomic relationship and everything. So based on the QSR, yes, I prepared my library and everything. But here I prepare some molecules and I check the biological molecules, and based on the biological activity, now I am designing the SAR. So here if you take these are my lead molecule. If you take this n substituent, we also tried with the aliphatic and aromatic uh, and like benzyl type of molecules. So if you go the aromatic, the activity is the more with the different substituents. Like if you go for the sorry, I forgot to keep the numbering. It's like 1 2 3 4 5 6. So based on the substitution, like 4-methoxy showing the higher activity when compared to 2-fluoro, and 3-methoxy and 4-fluoro, 2-bromo, 3-bromo, like the this is the different type of the activity what I'm showing based on the molecules. So we conclude that aromatic has the higher property of activity when compared to the aliphatic and benzyl groups. So now I'm going for the next step. Like till now what I showed you is the like only what we call as like one of the example. Now here my like the real work starts from here onwards. So after the census, I told you people the MTTSI should be the like I based on MTTSI, I identify some molecules and based on the targets. And after that, now I'm going for the in vivo targets. So like actually this is a clinical test. If we are starting from the these are called the preclinical and the called the preclinical. So now once is the mice model is successfully like passed, then we go for the clinically. Right now, my molecules, what I'm working in the Ohio State University is past the every molecules and the molecules are in the like in clinical trials. These are the not molecules what I shown you earlier. The molecules are different. Right now, the work has undergone like under patent, so that's why I can't be sure reveal those molecules. That's why I give the few of my old molecules and how I design the molecules. So here, as I mentioned you, it passes the cells and it passes the like in vitro and in vivo targets. And right now, my molecules are done phase zero studies. In the clinical test, how do we go for the like all you know phase zero? What is phase zero? 1, 2, 3, 4. Like for phase like phase zero, we go only like 10 to 50 like participants, and that too we have they have diseases, and we introduce our molecule to their body, and we take the blood samples and we check the thing. Like phase one is for the number high, like the thousands, and like phase two is the 10,000s, and phase three is the like both normal patients and disease patients, and phase four is the like final one, and post marketing is after prescribing the molecule, after prescribing the drugs to the patients, and is there any side effects or something. So we have to identify the everything. So here we already take the 80 subjects from the Oxford University and 80 from the Ghana University, and what are the molecules we synthesized, we give to the subjects. So after the subjects we get the repetitive time intervals like the 7 days, day 0, day 7, day 14, and we take the blood samples from the those patients. So after that, taking a blood samples, and we centrifuge the blood sample. So like we add we add some methanol to the blood sample to precipitate the proteins. Like if the blood has a lot of the proteins, hemoglobins and everything, so which will interfere the my like desire of the molecule. That's why we prepare those molecules, and after that we centrifuge those molecules. So we centrifuge the molecules, and we take the serum and plasma, and we take the plasma of the molecules, and after the plasma, we go for the uh, extraction of the metabolites from the plasma. So this is the different task. Like we added different type of solutions, and because after that we finally go for the metabolomics of the blood samples, because how the drug is metabolizing inside the body, and the based on the metabolism we also check the different aspects like bioavailability, bioaccessibility, T of life, and mechanistic pathway, and side effects, dose optimization, and selection of the patients, like healthy versus the disease, this thing. So here I selected the patients, like already diseased patients, how it working. So already in the animal studies, like in the for example, in the clinical studies, I go for the different concentration. I told you, like I go for the 0.5 to 5 microliter. In the mice studies, based on the body weight of the mice, we need to optimize the drug. So the drug the weight what we optimize over here, we transfer the same type amount to the patients also. But here also what happens, sometimes the dose will not efficient for the humans. So there was we need to like in the not the every time or every case, sometimes or some cases we need to optimize the dose over there also. And the side effects, like although I'm giving to the drug to the disease person only, but like is getting any side effects, like for example, like although if you like use the chemo drugs for the cancer, you get the like alopecia as a side effect, because the molecules what I'm preparing is only for the helping for the like rapiding dividing the cells. Like in our body, the rapid dividing is the hair, so it will be grow rapidly. That's why we used to go for haircut for the every week or some every month or something. That's why it will go and block the hair cells. Similar way, apart from it also go like how it affecting to the body. Like we diagnose the diseases, all the diseases from the person, and apart from cancer. So based on the our drug treatment, we need to check whether what about the remaining parameters of the body, like the heartbeat or hypertension or diabetes or like kidney chronic disorders and the fatty liver, what are the type of parameters we need to observe each and everything. And the mechanistic pathway, So I am giving to the body. I told you it will go different genes of the body and the thing, but the in the body we don't know like how it passes to the blood or to the oral or like it will go and how it would affect on the body. So we check the thing, and we need to check the bioavailability and bioaccessibility. Bioavailability means how much of drug is available inside the body. Like I am giving the three, for example, 3 mg of drug to the body. So among the 3 mg, it is not necessary go and take the all the 3 mg in the body. Sometimes it will be like 1 mg, 0.5 mg or depend because like the conjugation, what are the like the flow solubility or like lipophilicity based on the drug properties, the property will be the vary from the drug to drug. And bioaccessibility, although there is 100% bioavailability, there should not be like 100% bioaccessibility. Like bioaccessibility means how much the drug the body is accessing the drug, although available like if you give the IV, it is the bioavailability is like 100% when compared to the oral, but the bioaccessibility should not be 100% as like bioavailability. So we need to check that thing also. These studies we will do by the dissolution property, like in the labs we go for dissolution properties, in the body we go for the instrumentation property, like in the blood we go for the instrumentation. And after that, T of life, like how long the drug is staying in the body and eliminating in the body, because if long term staying in the liver and it also cause the infection to the liver or like accumulation of a drug, and the liver is, because liver is the main part to work remove the toxins from the body. If it is going to block the liver, so it will again the effect and everything. That's why we used to get the every sample, like we used to get the blood sample, urine sample, feces sample. So few of my colleagues are working on the feces and urine sample. I'm working on the blood sample. So we take the blood sample. We took the blood sample. I told you how to collect the plasma from the thing. After the plasma, we extract the proteins and we we extract the proteins and we collect it in the test tubes. And after that, we passes to the SPE filtration. This is like a type of a column. So if he passes over here, here's something like C8 sorbets are there, so it passes the only low molecular weight. I told you my drugs are like 500 Daltons something, less than 500 Daltons. But if you take the hemoglobin or blood sample, that is like a huge Daltons. So I don't want to all these things. I don't want to mess up the each and everything. So that's why I choose the only few of the molecules what I want, and I take these molecules and I dry the solvent, because this solvent we used to develop a method for the analysis of the blood and everything. So we dry the sample and we reconstruct the sample with the desired solvents, and we go for the LCMS metabolomics. So in the metabolomics, here I'm not showing any data because I told you these are under like patent. That's why I keep everything confidential. So in the metabolomics, for example, my parental peak is like 457, for example. So after that drug metabolism, it will be around like 127 or 300 something, whatever the thing. So now I should understand like how the drug is there and how it is breaking, and how which form is available in. Like some of the drugs are the because of low solubility and low thing, it won't be the available to the body. So in such a cases, we go for the like salt formation and we modify the drug, and we again go for the ingestion of this thing. And one more thing, and the second thing is like for example, few molecules are the getting like toxicity. Like example, having the nitro group and this type like trinitro, this type of the molecules show how much of toxicity. So to take off this toxicity, we modify the instead of nitro, we reduce the into MN or something, and we check the activity again. Some of the have trifluoro is a very good activity but against the toxicity. So apart from the trichloro, we go for the OCF methoxy OCF3 or only CF. So we go the different type of alterations and we see the modifying the molecules. And based on the metabolite, we go for the metabolomic studies, how it's affecting. And meanwhile, we also checking like in the blood, if we are giving 3 mg of dose in the blood and the LCMH, how much mg I'm able to see. So based on the thing, we identify the T of life and the biaccessibility of the like based on the T of life, how much how much it is showing inside the urine, inside the blood. So if it is showing the like if it is eliminating of 25% of blood means, or like the bioavailability maybe 25%, or it will eliminate from something else like the feces or like the urine. So we have to go for the data of each and everything based on the data, we conclude that how it is going and admitting the thing. So this is this is for the clinical and preclinical data how we used to do. So right now, like our drugs are in the phase 0 studies. So we got the blood samples and we also check, and few of the drugs are showing the really very good activity, and I am waiting for the rest of the patient samples. So like we have the, like in the clinical studies we have different criterias like the age criteria, sex, and disease and gender and everything. So we are taking the different type of blood samples, and once we get everything, and we go for the conclude the data. So as I showed the previous work, we published in the ACS Medcom letters. So that this is the work which I shown you before the preclinical test. Once the clinical stress, I told you both are the same molecules. The molecules are under the work is under this thing. So once it's done, I will good to go for the thing. And thank you. If you have any doubts.
Thank you, sir. Dear participants, if you have any queries, sir is free to ask. Sir, Balu sir is here for you.
Okay.
Okay, sir.
Hi, sir.
Sir, how are you?
Sir, doing good, sir. How are you? Balu sir is my BPA faculty. He is the one who teach me statistics and everything. Hi, sir. How are you, sir?
Fine, sir. How are you?
Yeah,
Where
I'm
are
doing
you?
good, sir.
Okay, okay. Your section is very well, very good, sir. Our
Okay, thank you,
faculty
sir.
said that they gained a lot of knowledge by hearing your section.
Thank you very much, sir.
Okay, sir. I will call you.
Yeah, definitely, sir. Okay, sir.
Thank you, sir.
Yeah. Okay, sir. Anyone have any doubts? So, like, if you're not getting doubts, means like two things. One is you didn't understand anything, or you can understand like everything. So I don't know.
Okay, sir, thank you. I
Yeah,
think so
okay,
there
ma'am.
were no questions. You have explained in such a beautiful manner. So, concluding our section, thank you, Dr. Y. Prasanna Anjaneyulu, sir, for engaging and highly insightful lecture. On behalf of BV Institute of Pharmaceutical Sciences, we express our deep gratitude and present a token of appreciation in the form of certificates. Sir.
Thank you, ma'am.
सबसे वही ये करे वो करे ये करे वो करे वही चल रहा है
1 minute.
रुद्र सर ने बोल दिया कोई भी कर लो मैंने भी बोल दिया कोई भी कर लो
Sir, on behalf of VV Institute of Pharmaceutical Sciences, we express our deep gratitude and present a token of appreciation in the form of certificate. Sir, thank you, sir, for your
Thank you very much, ma'am.
informative lecture.
Okay, ma'am.
Thank you, sir. You can please.
Yeah, thank you, ma'am. Bye, ma'am.
Dear participants, the attendance form will be shared in the chat box. Please ensure to fill it before leaving the section. Dear participants, the attendance form has been shared in the chat box. Please ensure to fill it before leaving the section. Thank you all, and we look forward to meeting you in the final section tomorrow. In the final section, we would like to share the feedback form as well as the assessment form. Thank you all.